So, I mean, I want to start with the last point from the previous talk. I really don't know how much Konorski is now. I can tell you that I know him, that I know much of his work, and I know many people that know him. And I cannot tell you much about Konorski. I mean, I'm too young to know him or to know much about him, but I came here basically to learn more about him. I mean, out of curiosity of somebody I consider brilliant, who has brilliant ideas. And the idea that comes closer to my work, or the one that most strikes me, is the one of Gnostic neurons, which was mentioned in the previous talk. And I think Konorski was brilliant in describing that, in thinking, well, we actually don't think about Gabor patches that we use in experiments or oriented lines, as Hubel and Wiesel had, of moving dots or so on. We just see people. We see chairs, desks, computers, faces we know. So these are our unitary percepts, as I believe he called them, and we may have neurons that actually do represent these unitary percepts. And this is what he called Gnostic neurons, or intelligent neurons, if you want to take the translation literally. So I will basically tell you about something we found in human brains, in humans, which we call concept cells. And this is, I mean, we published the first paper in 2005, and these are recordings done in humans. And we will discuss, if you want, a little bit how close these concept cells come to this concept of Gnostic neurons. So, but let's, I change a little bit the order of my slides, and let's talk a little bit about this history of Gnostic neurons. And this goes all the way back, surprisingly, very, very surprisingly, and this to me is quite striking, to William James. William James wrote a book in 1890 called Principles of Psychology, which is more or less the basis of psychology, modern psychology, 1890. And in 1890 he talked about nerve cells that may encode, I mean, particular behavior, single nerve cells. So he described this just before Cajal neurons doctrine, which was at the end of the century, more or less. So even before Cajal, William James was talking about nerve cells that may encode behavior. Sigmund Freud, I mean, we know him about psychoanalysis, but he had also very good insights in neurophysiology, and he also talked about, what is that, also at the end of the 19th century, about psi and phi cells that were supposed to be linked to different states and behavior. Now, this view of William James was somehow demolished by Charles Harrington in the 1940s in his book, and he took this idea of James and he described, well, I mean, he said, well, pontifical neurons, like you have a single pope being deciding behaviors, and he completely disregarded this idea, and he said the brain instead works as a billionfold democracy. So it's basically there are large networks of neurons doing everything. They're not single neurons with such very high level specialization. But Jerzy Konorski actually disagreed with him, and in his book, as you heard in the nice previous presentation, I mean, he was describing Gnostic neurons, and it was not just an idea. I think he had very, very good insights for thinking that there could be neurons like that. First, he knew about pathological problems, like different type of agnosias, and he said, well, agnosia is not that you don't see an oriented line in some orientation. The agnosia is that maybe you don't recognize certain categories, right, in animate things or inanimate things or so on. So there must be neurons that are specialized. And most importantly, he knew very, very well the work of Hubel and Wiesel, and I come to this in the next slide. Now, the one that somehow popularized this story of this whole thing was Jerry Letwin with his very famous story of grandmother cells, right? So he was just putting in the name, and this somehow caught up in the field. Well, do we have cells firing to grandma? Do we have cells firing to, I don't know, my wife, my children, and so on? And people have been discussing this since the 60s. And another person I would like to mention is Horace Barlow, who in 1972 wrote a very groundbreaking paper in perception, and there he talked about cardinal neurons. He said there won't be a single pope ruling behavior, but there may be something like a college of cardinals. And it will not be a millionfold democracy, right? There will be specialized neurons, but there won't be just one. So let's do a little sketch of one slide of visual perception, but I want to bring this idea. At the back of the head, I mean, Hubel and Wiesel describe these neurons that in cortical area B1 that respond to different local orientations, and this is what we call simple cells in B1, right? So these cells will fire to a vertical line at a particular location, but not to an oblique line or an horizontal line. Now, I don't have this in this slide, but there are also complex cells with they fire to, for example, angles, to something a bit more complex. So Konorski knew about this very well. So he knew that there are simple cells, and these are supposed to feed into complex cells. Simple cells will fire to oriented lines, complex cells to joint lines, and then he wondered, well, maybe this hierarchical organization goes forward, goes further, and then you have more and more complex cells, and at the end of the day, you end up with these Gnostic neurons, right? And this had a huge impact on somebody, and I just know this story from reading the papers of this somebody, and this person is Charlie Gross, and he writes in an article in the Neuroscientist. He described how, I think he was visiting here, where he was visiting Konorski in the 60s, and he described how this changes his scientific career. And basically, Charlie Gross took this idea of having neurons more and more specialized and went back to MIT and tested it, and he found neurons in inferotemporal cortex at the end of the visual processing pathway, ventral visual processing pathway, that actually do fire to these more conceptual things, like, for example, this neuron here firing to faces, like a monkey face or a caricature face. He found also neurons firing to hands or to body parts and so on. And Charlie Gross describes this himself. I mean, he says he went for this experiment following Konorski, right? And actually, Charlie Gross was one of the people that wrote the review of Konorski's book in 1968. So, and we know this since the late 60s, right? That we have these very specialized neurons, and then we know about this ventral visual pathway from V1 to IT, and in V1 we process very local details, in IT we process something more complex. In the 2000 Winrich Rival with Dorit Sau also described what we call face patches now, so they're very specialized areas around the temporal lobe that they fire to faces. Now, this area IT has massive projections to another very interesting area called the hippocampus, right? And all what I will talk, I mean, in the remaining of the talk, will be of neurons recorded in this area. The hippocampus, and I would say the surrounding cortex of the hippocampus, entorhinal cortex, amygdala, and parahippocampal cortex. And I will be basically telling you about recordings in this area, but not in monkeys, I mean, as Charlie Gross, or in cats, or monkeys later, as Ruben and Vincent. This will be in humans, right? So we have single cell recordings in humans. How come we can record single cells in humans? Well, these are patients that are candidates to epilepsy surgery, and surgeons and team of epileptologists try to localize as best as possible where the seizures come from to try to remove surgically the area starting the seizures, if it is possible. And sometimes to pinpoint exactly where the seizures come from, they will put intracranial electrodes. And this is the surgical procedure. They have, the patient is there, has an stereotaxic frame, and then they will put electrodes, they're little needles, maybe 1 millimeter thick, and they will target the areas that they believe are starting the seizures. One of the areas that is the usual suspect for starting the seizures is the hippocampus. That's why many times they end up putting these electrodes in the hippocampus and the surrounding cortex, just to try to find out where the seizures come from. And this is work I have done in collaboration. I mean, we still continue working together in many things with Christoph Koch at Caltech and Itzhak Fried at UCLA. The recordings I will show you were taken actually at UCLA, and Itzhak is the head of the epilepsy there, and also doing cognitive neuroscience. I mean, and basically what Itzhak developed with his team of people at UCLA is a way of recording single cells from these intracranial electrodes. And the trick is that if this is the intracranial electrode they typically use for recording where seizures come from, Itzhak managed to have a way to have microwires at the end of the intracranial electrode, and then from these microwires we can record single cell activity. These microwires are about 40 microns thick, so it's smaller than 1 hair, much smaller than 1 hair. So all what I will show you is data recording from these different microwires. So now, I go back to the ideas of Konorski, and I want to explain you where we are. I think you saw this picture many times, and basically whenever somebody shows this picture, it's just to tell you how complex the visual system is. In my case, when I want to tell you something different, I basically am saying that I'm recording here, right? So this means these neurons are not magical. I mean, these Gnostic neurons, if you want to call it, or concept cells, they don't have any specific properties that make them so different from others. No, they're the same as others. They're pyramidal cells as the ones you find in mice, actually. The difference is that these neurons are receiving heavily processed data, right? So the output that you see from this neuron has been processed by several other areas before, right? So what do these neurons do? That's my favorite one. This is the one firing to, I guess you know Maradona, an Argentinian soccer player. So there was a patient that liked football, and I showed pictures of football people, and in particular about Maradona. And this is actually the raw trace, high pass filter. I mean, this is the only thing I have done to the data. This is the raw data coming out of one electrode. And you see, I show a picture of Brad Pitt, and this is the time I show the picture, the neuron does nothing. I show a picture of Maradona, the neuron likes it and fire. I show a picture of another sportsman from Argentina, basketball player, Manu Ginobili, the neuron doesn't like it. Another picture of somebody the patient knew, Robert Plant, the singer of Led Zeppelin, it's a rock band, the neuron didn't like it. A picture of Maradona again, the neuron liked it.
But would the face of Maradona be the same?
I will come back to this in a minute. That will be the question. Actually, this is first. I mean, why do we use this picture first? And I mean, I confess, it's fun. I mean, we like showing these things. But second, we always try to show things that are very familiar to the patient, because the chances of getting responses are much higher than if we show something else. The patient knew football very well, was a fan of Maradona. The patient was a big fan of this singer, the band Led Zeppelin, so he really liked this band. It's not just emotion, but we usually tend to show things that are very familiar to the patient, because then we have a higher chance, much higher chance of getting responses, and we show this statistically, right? Now, I will come to your question in a second, but first let me tell you about something. There's a problem here. This is very cool, this is very nice, but there's a problem because I did like a half-an-hour experiment, right? And the neuron fired to Maradona and fired to absolutely nothing else. Now, there's a technical problem of getting a neuron as clean as this one. This is not typically the case. This neuron is masked by the activity of other neurons, because when you put an electrode, you don't record from a single neuron. At least in chronic implants, you record from many. So you have to do something called a spike sorting, but that's a technical detail. Don't worry about this. This we can do. But the second point is, if I wouldn't have shown the picture of Maradona, I would have missed this neuron, right? So if in my data set of pictures that I show in my laptop, Maradona wouldn't have been there, the neuron was there the whole time, but it would have never fired, because they tend to be silent if you don't show the right stimulus. And this is another case. This is another patient in the hippocampus, and I don't know if you can see it, this neuron fired very strongly to the map of Italy, just the contour map of Italy. And it went from, this is the time of presentation of the stimulus, is 1 second presentation, 1 second before. It went from complete silence to up to 40, 50 hertz. It's very strong fire. Now, same point. If I wouldn't have shown the picture of the map of Italy to this patient, I would have missed the neuron. So how can we cope with that? Well, we decided to change the experiments and to do the experiments in two ways, in 2 sessions, right? So first we say, well, let's go to the patient and show as many pictures as we can, and try to show things that are familiar to the patient, that the patient knows well. Then we analyze the data, and then we decide what to do to follow up this experiment, right? So first experiment, we call it screening session. We show many pictures and see what neurons do, if there's any neuron responding to any of the pictures we show. I'm going to show you one example of one of these sessions. This is in one patient in the hippocampus, same patient of the map of Italy. And this neuron, we call it AM session because we tend to do it in the morning. It's not fired to all these people, to all these places, a basketball player and so on, faces and so on, but it fired very strongly to her. And that's Jennifer Aniston, the actress, right? And that's what people call the Jennifer Aniston neuron, because it was actually the first one I ever saw. I mean, the first one fired to Jennifer Aniston. Now, your question. Well, is a neuron firing to this specific picture of Jennifer Aniston because of some particular trivial reason? Like, for example, this is the only one that has a blue background, right? Maybe the neuron is firing to blue, or maybe the neuron is firing to something a little bit more complex, like the contrast between her hair color and blue, I mean yellow against blue, or something like that. Or is it really firing to her, to Jennifer Aniston? Okay, the idea is that we do the experiment, we get the data, analyze the data as quickly as we can, and then based on these results, we do the follow-up experiment. And the follow-up experiment here, to answer this question, is very simple. We get into Google Images, we got at the time different pictures of Jennifer Aniston, different pictures of different people, and repeat the experiment. And then the question is, or was at the time. Other cells firing to visual features or to the identity. They're firing to Jennifer Aniston. So we repeated the experiment, and now showing many different pictures, as different as possible, and this is what we got. Same neuron, a bit after, right? The PM session. So the neuron is not firing to different people. Another picture with a blue background, the Eiffel Tower in this case, not firing to these faces, another blonde actress, nothing, but it's firing to 3 different pictures of Jennifer Aniston. And you can do statistics in many different ways. This is highly, highly significant. I'll show you more responses. In this case, I show 87 pictures altogether. I mean, we cannot show too many pictures because our time with the patient is limited. We try not to go over half an hour experiment. So the picture fired to another, the neuron fired to another picture of Jennifer Aniston and another one, and it did not fire to any of many pictures of Julia Roberts, which, I mean, I use as control, or other people, right? It did not fire to another person she really liked, Kobe Bryant, the basketball player of Los Angeles Lakers at the time. I show you more. It did not fire to different people, and it fired to Jennifer Aniston again, and to Jennifer Aniston again. So 7 different pictures of Jennifer Aniston, as different as I could get in a quick search in Google Images, and the neuron fired to all of them, and this is highly, highly significant. If you do statistics, this is highly, highly significant. Now there was a curious thing here, which, I mean, people tend to laugh, but I found it very intriguing at the time. When I showed the time they were married, I showed Jennifer Aniston with Brad Pitt at the time they were husband and wife, and the neuron didn't like it. The neuron didn't like it. 3 different pictures, and the neuron did not fire, which is quite interesting. We can talk about this later because it will take me the whole talk to talk about this. It's quite interesting. You show Jennifer Aniston, no matter how you show her, the neuron fires, but you show her with Brad Pitt, and the neuron gets blocked. Right? So it's a different thing.
If you show Brad Pitt like in a paired pulse paradigm where he precedes her by some latency, will it mask the response?
We didn't do it here. No, that's the problem with human recording. Sometimes you can do many things, but we have very limited time with the patient. So there are many things. I mean, when I see the responses, there are many things like what you said. There are many things I would like to do, but we have only 1 week with the patient, and I mean, I don't know.
Did you try writing Jennifer?
I go to this in a second. I did try it and it didn't respond to writing.
Did you try to show Maradona picture without ball?
Without the ball?
Yes.
Yes, it did fire. I'm in England, so I always show this thing in England, and they hate it when I show Maradona. I always go, did you try Maradona hitting the ball with the hand? I said, no, he never did. So, okay, so let me move on. I show you another example. I mean, there are many of these neurons, many. I just show you a couple of examples, and I mean, we have many papers published. You can see tons of examples of that. But I'll show you another one that is very interesting. This is another actress, Halle Berry, and you see the response, very strong here to Halle Berry. And then we did the same trick. We analyzed the data quickly. We repeat the experiment a bit later, but now showing many different pictures of Halle Berry and see what happens. And this is what happens later, right? A couple of hours later or so, we repeat the experiment, we get the same neuron in the hippocampus. This is another patient, by the way, and nothing to all these people, very famous people, known to the subject, George Bush, Bill Clinton, and so on. And here is Halle Berry, Halle Berry, Halle Berry, 4 different pictures of her, and this is highly significant. The highly significant response, much higher than anything else. Julia Roberts, again, Julia Roberts poor, there's nothing for her. And at the time we were doing this experiment, Halle Berry was releasing a film called Catwoman. And you may wonder why in hell I'm telling you this. Well, it's because it's interesting, because she was all over promoting her film Catwoman, and she was masked, so you couldn't see her face, otherwise the film won't make sense, right? So, but we all knew that that's Halle Berry, because we all knew that Halle Berry is the actress of Catwoman. The patient knew that, and then we thought, well, let's show also Halle Berry dressed as Catwoman and see what the neuron does, and that was all in the same experiment. I mean, we planned it before to put this picture. It's not firing to all these people, the same neuron, no? And here is Halle Berry as Catwoman. We have 3 different pictures, and we have 3 good responses, as when you show the face of Halle Berry. So it's not the face that is triggering the firing of the neuron, it's something at the higher level. At the time we did this experiment, we were trying to play around with the limits of visual perception, and one way to play with this is using caricatures, because in a caricature you distort the shape of the face, but you can still recognize the person. Like, this is one of Michael Jackson, for example, one of Eddie Murphy, and here's the one of Halle Berry, and the neuron also fire. Okay, but now it comes to your question. I mean, I'm trying to prove that this is firing to the concept, right? It's not firing to any visual detail. But there's a very easy way to prove that because we're doing experiments with humans. And the way to prove it is what you say, we can just write a name. So we did that. And these are different names. All these pictures are intermixed, right? There are about 100 pictures. They're shown 6 times each for 1 second. So, I don't know, Kobe Bryant, Julia Roberts, Catwoman, George Bush, and look at this, Halle Berry. Whenever Halle Berry was written in my computer screen, right, the neuron fired the same or something, maybe a bit more than, I mean, compared to showing the picture or showing her picture dressed as Catwoman. So they're really firing to the concept. Now, what about sound? What will happen if I will say the name? Well, it doesn't sound very interesting, right? Because I already showed writing the name. But it is interesting because it's a completely different sensory modality. I mean, sound don't go through the ventral visual pathway. Sound stimuli goes through the ear. I mean, from the ear goes straight to the temporal lobe. Visual stimuli, I mean, for a reader string or for a picture, they go to, I mean, they go to the back of the head, and then they have a processing pathway which is different from letter strings than from pictures. But some stimuli, they have a completely different route, and they also merge and get into the hippocampus. So the question was, will neurons in the hippocampus, the same neurons, fire also to the same concept, but triggered by a different sensory modality? And after such a long introduction, I mean, you can guess that the answer is yes. So here is a neuron that fired to Oprah Winfrey, hippocampal neuron of another patient, fired very strong to Oprah Winfrey, I mean the presenter from TV, to the name written on the screen, Oprah Winfrey. Look, this is 0, this is baseline, and you write the name and the neuron fires. And this is just the computer saying Oprah.
Oprah.
So it's just my laptop saying Oprah, and that triggered the fire of the neuron.
Cameron Diaz.
So it's a computer synthesized voice. If the computer says something else, like Cameron Diaz, and we had many of these stimuli, the neuron didn't fire. I'll show you another one. This neuron fired, again, something that was very familiar to the patient, fired to Luke Skywalker, the character of the film Star Wars, right? So again, look at the baseline. This is the second before stimulus presentation. There was absolutely nothing. I show the picture of Luke Skywalker, there's a very strong response. 3 different pictures, 3 good responses. This is the name written on the screen, Luke Skywalker, the neuron fire. And this is a male voice and a female voice saying Luke Skywalker.
Luke Skywalker.
So that's the male voice,
Luke Skywalker.
the female voice, and you get the same response. So no matter how you say the name or how you bring the concept, the neuron fire. But look carefully, there's something even more interesting in this slide, right? Look at that. You know who this guy is? This is Yoda. Yoda is another Jedi from Star Wars. And if you do not know what a Jedi is, well, maybe you should get out of the lab every now and then. So these are 2 like fighters for the good side. I mean, for in this movie Star Wars. So they are quite linked, no? So the neuron is not just firing to Luke Skywalker; it's also firing to Yoda. I only show one picture of Yoda in this experiment. Now, this is not uncommon that we find neurons firing to more than one thing, and actually makes a lot of sense when I tell you what I believe these neurons do. So keep this in mind. Okay. Well, this is another very interesting neuron, and I say this very quickly. This neuron fired to Arne, and then you might wonder, well, Arne, is he a famous actor? Is he a famous politician or so? No, he's not a famous guy. Well, maybe he's a brilliant scientist, but I mean, very likely you won't know him. So Arne was a guy doing experiments with the patient. I mean, he was trying completely different things, and we got a response to him. 3 different pictures of him. His name, well, this is Arne, and his name saved by the computer. And why this neuron is interesting? Because the patient did not know him before. So Arne was new to the patient, and I'm pretty sure of that because we don't interact with the patient before they go to hospital, before they're in the ward. So I know that the patient did not know Arne 1 or 2 days before I did this experiment. So this means that this representation, this conceptual representation, can be created relatively quickly, within a day or maybe less. I mean, we know now with new experiments that it could take just seconds. It could be very fast. Okay, I skip all this. Now, back to the original question about Gnostic cells. I mean, do we have one neuron for Jennifer Aniston? This one here is Halle Berry, actually. So somebody did this poster a while ago, and I found it very cool. So do we have one neuron to Jennifer Aniston, one neuron to Halle Berry, and so on? are these what Jerry Letwin called grandmother cells, or are these close to what Leicerster Gnostic called Gnostic neurons? Okay, the problem with grandmother cells or Gnostic neurons is that we can discuss forever, and very likely we're talking about different things. So we have to agree on a definition. So let's go to potential definitions and go step by step, one by one. Grandmother cell, some people, very few actually, very few people will take grandmother cell as one neuron encodes for one and only one phase. So Jennifer Aniston will have only one neuron representing her in the brain. Well, this is impossible, right? Because if I do find a neuron firing to Jennifer Aniston, there have to be more. Because the chance that I will find the one and only out of billions is 0. I can record up to 100 neurons simultaneously, right? The chance that one of these 100 will be the only one among billions firing to Jennifer Aniston is close to 0. So if I do find a neuron firing to Jennifer Aniston, there have to be more. And we can do many statistical arguments and so on, but don't worry about that. Now you can say, well, maybe it can be that we have many neurons firing to one and only one concept, right? Well, this could be. It will be very hard to prove because I can show about 100 pictures. I cannot show every single thing that is around. It's impossible. But also, there's a way to disprove it. I do find in many cases neurons that fire to more than one thing. I already showed you the one firing to Luke Skywalker and Yoda. And this is not that rare. It is common that we find neurons firing to 2 different concepts, sometimes more. When I found the Jennifer Aniston neuron, I repeated the experiment the day after, and then I show many different things related to the TV series Friends, because that's what made her famous. Jennifer Aniston started to be famous because of the TV series Friends. And the neuron actually fired very strongly to this woman here, and this is Lisa Kudrow. She's a co-star or she was a co-star in the TV series Friends. Now, maybe now I confuse you a little bit, because you're thinking, well, are these grandmother cells, are these Gnostic cells or not? Depends on your definition. But what I want to say is I want you to understand very clearly what these concept cells, as I call them, are. They fire to concepts. They do not fire to details. They do not fire to Jennifer Aniston because of this particular picture, or because of the background, or the hair color, or because she's looking in front of you, a profile. No, you show her and the neuron fires, right? So they fire to concepts. They fire to very, very few things. If I show 100 things, they will fire maybe to 1 or 2, but not to 50 things, right? So they're very sparse. It's a sparse representation. And third, and most importantly, if they fire to more than one concept, these concepts are always related, right? Luke Skywalker and Yoda, Jennifer Aniston and Lisa Kudrow, and I can tell you tons of examples like that. Neurons firing to more than one thing, but these things are always related. So what are these cells doing? And why do we have neurons firing to related concepts? Well, if you want to understand what these cells do, I mean, we cannot ignore the most brutal piece of evidence about hippocampus, and this comes from patient HM. And patient HM, as you all, I guess you know, had his both hippocampi removed, in a surgical procedure in the 50s, tried to cure him from epilepsy. In our days, we never remove both hippocampus, we remove just one and leave another one. And you know the story of patient HM, no? I mean, when they removed the hippocampus, they realized that HM could not form new memories. He could still have memories from the past, I mean, with some degree he was not perfect with that, but he could still have some memories from the past. He had no problem in visual recognition. So this means these neurons are in the hippocampus and the surrounding cortex. And we know that the HM didn't have hippocampus, and HM could recognize people, could recognize things. So we know that these neurons, these Jennifer Aniston neurons, are not involved in recognition. We also know that these neurons are not the final storage of memory, because HM could still remember things from before the surgery. He knew who his mother was, who the surgeon was, and so on. So, but HM could not form new memories. So my postulation, my hypothesis is that these neurons are involved in creating new memories. So, and I like to call them the building blocks of memory. So they're basically a semantic representation of concepts that you have to make links. And episodic memories, making links, making association is like the basis of new episodic memories. 1 year from now, I remember that I have been in Warsaw and I gave a talk. I remember the Nencki Institute, I talk about Konorski, and I talk about the Jennifer Aniston neuron. So we always abstract things, have concepts, and we make link between concepts. And this is the way we tend to store things in long-term memory. So no surprise, you have such conceptual representation in an area that is dedicated to memory. And no surprise, we found neurons encoding things that are highly related. Well, these neurons are encoding associations, right? Because association is a key, feature for memory. So we call these like the building blocks of memory. It's like the Legos that you may put together to form new memories. So I finish by saying that concept cells have an abstract, right? Conceptual, explicit, because a neuron is telling you what the subject is doing or thinking, and sparse representation of the meaning of the stimulus, right? Whatever triggers the concept, the neuron will fire. And we believe they are the building blocks of declarative memories, particularly episodic memories, and is what is linking perception and memory functions. Okay, I think I finished there. Thank you.